it is my privilege and pleasure to introduce Dr. Martin Gaudinski to you. Dr. Gaudinski is well known to all of us. He's a double Hoyer, having gone to medical school here with us and also having done his residency here. He also moonlights here as a hospitalist. After he finished his fellowship, he went to the NIH, where he did his fellowship in allergy and immunology, and then stayed on at the NIH to implement the first in-human clinical trials and passive prevention monoclonal antibodies against infectious diseases and other priority pathogens, which is what his grand rounds topic is all about. Thank you very much, Dr. Gaudinski, for taking the time to speak to your Georgetown family again.
Thank you, Dr. Kumar, and it truly is a pleasure to be back, and thank you for the kind introduction. As I was preparing to talk today, I was dropping my son off at school, and I said, Benjamin, say a prayer for me today because I'm giving a talk. And he said, in his form of a prayer was, don't mess up. So I'm hoping that I just won't mess up today. And then in a show of goodwill, he asked me what my topic was, and I told him, and he said, so that's simple, you'll do fine, which was also reassuring. So hopefully what I'll be able to do is fulfill that promise of making a topic pretty simple and tell a couple stories along the way. So these are the objectives for the talk today. And I put on my disclosure that I have accepted full-time employment for GlaxoSmithKline beginning next week. So this is actually my final week at NIH and in uniform for the Public Health Service. And I don't think that will matter here today because none of the things that I'm going to talk about have anything to do with GSK. Now, as Dr. Kumar mentioned, this is somewhat of a homecoming. So I wanted to think a little bit about, how did I take Georgetown from the campus into what I do on a daily basis? And I really think that what I do has a lot to do with the cura personalis model. And so I really just wrote down that the part of the purpose of the talk today is to persuade you that the type of biomedical research I do is an extension of caring for the whole person by caring for whole populations. And it differs from caring for a patient. It may come to nothing at all in the end, whereas caring for a patient always comes to something. But for many of the people, perhaps trainees that are here, or some of the people who trained me, just to get a sense of how that cura personalis goes out into the world. And then more additionally, that the type of clinical trials that I'm doing are specifically designed to benefit people in low and middle income countries, and that there's a certain sense of accomplishment in being able to push forward something that's going to be helpful for other people, not necessarily on our own soil. So I thank you for the attention to all of that. And the remainder of the talk will be a little less preachy than that homily I just gave. So my general impression is that monoclonal antibodies to treat infectious diseases or prevent them has come to people's awareness greater now because of the monoclonals that have been employed against COVID-19 and the virus SARS-CoV-2. Now, this talk really attempts to make that perhaps black box a little more understandable. And one of the demonstrations I'd like to make is that understanding the underlying biology of a disease is necessary first prior to designing any monoclonal antibodies that may be beneficial. Now, also, when feasible, active immunization with a vaccine, I think, is the superior option. It tends to be more effective, it tends to be more durable, it tends to be more cost-effective, all things that I'll talk about later on. But sometimes it's just not possible. And passive immunization is an alternative when immunization is technically challenging or there's a virulent disease process where you're essentially having the immune system playing lightning chess with the pathogen and it may not win. And so monoclonal antibodies that can be produced ahead of time can be produced from vaccinated or convalescent individuals, And really, you're looking for an exceptional immune response and to duplicate that for the greater population. And I'm going to talk about Ebola and HIV en route to getting to malaria, which I hope to draw out as a particular pathogen of interest. Now, the dog here is Balto, and Balto has a statue in Central Park. And he was actually a sled dog that was able to take diphtheria antitoxin to Nome, Alaska in the early part of the 20th century during the middle of a diphtheria outbreak. And I bring this up to say that what we're doing is actually not new, that passive immunization has been going on for a long time and was actually the cause of one of the first Nobel Prizes. And so this is an extension of the history of where we've been and really what we're doing in the medical armamentarium. Now, if there's a punchline to this talk, it's malaria. But in order to understand that joke, it may be helpful to understand other pathogens. And this relates back to the underlying biology as something that needs to be accepted rather than fought against. And ultimately, you won't win if you're trying to fight nature. And vaccinology in general is really just assisting nature and not commanding it. And so my first example is going to be Ebola and the natural history of Ebola infection, which is seen on this slide. And if you look here, this is just, you get infected with Ebola, and then what happens? What does the immune system seek to do? Well, your innate immune system with all of the components of macrophages, natural killer cells, certain types of T cells that you might have, they're all going to go into effect. And then you have the virus, which begins to spike. And then with the advent of the virus, your B cells will start to create their antibodies. First, they create IgM. You'll have a T cell response that goes up, and you'll have an IgG production. And this actually works very well. The problem is you die well before this comes to any sort of a productive response. Observed death tends to be within 4 to 16 days after the infection occurs. And your IgG response is going to occur, like most IgG responses, about 3 weeks after the pathogen presents itself. And so what we need to do is we need to learn from this and take this right here and give it to somebody here in order to present a productive response prior to their death. And in that case, the mAb is acting like a treatment. Now, what about this? The intention is to create an intervention, but that's really only the first step. It's critical to understand the biology of the underlying pathogen and then understand the process with what you can do to make a mAb. So on the left, you're going to see how Ebola works. And this is actually similar to how malaria will work that I'll describe in the future. And so you have Ebola, which is a long filovirus, and all of this blue dotting it is glycoprotein. And the virus goes onto the cell, which is the gray component here. It gets taken into the cell by macropinocytosis, and then it goes into an endosome. And you can see that the glycoprotein here has these blue features of it in dark blue. And then in the acidic environment and through various other aspects of cathepsin cleavage, you have the glycoprotein cleaved, which leaves a receptor binding site, the RBD, and a receptor binding domain, which goes on to an MPC1, and then that's where you get viral replication after it leaves the endosome. And so this is the underlying biology. In order to create a monoclonal, you have to figure out whether a monoclonal is effective in this process. Many of the antibodies are going to bind, for example, to this blue component that ends up getting cleaved off. And if so, then it's no longer biologically active. And so what you need to do is you need to find an antibody that actually stays after this cleavage event and can prevent the binding to MPC1 in order to prevent all of the downstream damage. And we were able to find an antibody like that. And this is how you do it. You find an Ebola survivor, for example, or you can use a genetically modified mouse. You isolate the cells that create the immune cells, so B cells. You isolate the actual gene encoding the productive antibody. You put that gene into other types of cells, such as Chinese hamster ovary cells or CHO cells, and they're going to then use that gene to produce the antibodies for you in a fermenter, kind of like you're making beer at home. It's very similar. And then you purify that, you vial it, and then you use that special antibody in animals and then humans for the first time. And then if all is well, you administer it to Ebola patients in a clinical trial. And all of that is how you are going to, in lightning speed, try to figure out in the middle of an outbreak whether you have a monoclonal antibody that's actually productive. Now, this is what we have done in the creation of mAb 114, which is now under the FDA-approved trade name Ebanga. And this is a monoclonal antibody to treat Ebola virus disease. And we had, for a long time, been trying to create such a monoclonal antibody. We created it and it was ready for testing. But then the 2018 outbreak of Ebola in DRC occurred. And in rapid time, we did a phase 1 trial on the safety and tolerability and pharmacokinetics of the monoclonal antibody, which allowed it to go into an efficacy trial that was run by NIAID and Dr. Lane and others that was then published. And it showed that, in fact, 2 treatments, mAb 114 as well as a Regeneron product, were capable of preventing death. Now, it wasn't perfect, but it was an actual clinical trial data done in a rigorous manner that was able to show that we had benefit from a monoclonal antibody. And in many ways, this was a dress rehearsal for COVID-19 and a lot of the vaccine and monoclonal antibody trials that were done really in record speed. And what are the lessons from this? Well, one of the things I'd like to highlight in why Ebanga was successful is because Ebola itself is susceptible to antibody-mediated immunity, and not all pathogens are. And the technical features of mAb 114 exploit a unique weakness of Ebola, so that it can bind and coat the virion with all of those glycoproteins. It can stay in the acidified endosome environment. It can survive the cleavage event, and it can remain bound to prevent the Ebola receptor binding domain to binding to MPC1 to prevent infection. And so, you know, it's not perfect. Mortality is still high in late infection and high viral load settings. But when the goal is prevention and not treatment, the rules of mAb design also hold. And so what I've taken from the Ebola experience, I'd like to put into the HIV experience, which is admittedly a very different pathogen, which I respect to a very high degree. So pivoting from the success of Ebola, going to HIV, where honestly, we're not yet at that point of success. And it's not exactly clear what the path forward to any success with HIV is. I would say that we're still in that initial phase of trying to understand the underlying biology, to understand further how that intervention moment can happen with an antibody. Interestingly, this struggle with HIV, and it is a genuine scientific struggle, has been very fruitful to address lesser pathogens, easier to handle, like Ebola, like malaria. Where I am right now at NIAID, the Vaccine Research Center, we were actually founded by President Bill Clinton in order to come up with an HIV vaccine. And we have not done that yet, but the struggle has allowed us to create advances in structural biology to understand viral structures, in order to understand different types of vaccine platforms, and to explore different types of monoclonal antibodies in such a way that we've been able to come up with successful or clinically advantageous Ebola therapeutics, both vaccine as well as the monoclonal that I talked about, to talk about SARS-CoV-2, to talk about malaria, to talk about chikungunya, to talk about respiratory syncytial virus. There's a lot that has been informed by the struggle of HIV. But why is HIV so hard to deal with? Well, it has a multitudinous mutations that I think we're all aware of. It has the speed of integrating into the host genome, which is a problem. And I don't know that it's as recognized, but the glycosylation that shields the actual protein structures of the virus that antibodies would bind to is immense. It's light years beyond something like a virus, like respiratory syncytial virus or other features. And so HIV is a very, very difficult task to handle from the vaccine or the antibody perspective. And yet, contrary to some thoughts, antibodies, I do think, will play a role in ultimate vaccine design, as well as broadly neutralizing antibodies. And I'll talk about some of that experiment of nature here. What you can see is that on this slide, most people, when they encounter a virus, they have high degree of viremia. The virus begins to mutate. You can see that in the different types of colors of virus. And you have moderate to high viremia, and this goes on for a great number of years. But there are a separate group of people who are slow progressors, who they have their initial virus, they have some low viremia, but it's present. And they have antibodies that develop to the point that they can block this initial virus, and it stops its mutation path going forward because what's actually present gets handled by the immune system. What you can see in general is you have low affinity antibodies to HIV, and then over the course of months, you start to get more and more and more antibodies that can actually handle HIV to a very high degree. The problem is it takes years to develop, and by the time they do develop, the person is already infected. And so you're at a point where it doesn't help them. But what if you could take these very good antibodies that are produced by a few elite patients and give them to people who are not infected, but at risk of being infected, and then you can prevent their infection going forward. Now, this has been done for a number of broadly neutralizing antibodies, or bNAbs, and for a number of different sites of vulnerability on the HIV-1 viral spike protein. And so here you can see an example of that. You have a CD4 binding site. So this is where the virus will bind to CD4. And you have a number of antibodies that will block that event, VRC01, named after the Vaccine Research Center, I'll talk about extensively, as well as VRC01LS. But there are others. You can have a glycan sites that are targeted by antibodies. You can have the EMPER, or membrane proximal external region, different areas, and you can even create combinations. So you'll have a tri-specific antibody in which it has components that bind to CD4, V2, and the EMPER region all at once in an effort to have a very, very broad response. And so I am going to talk about VRC01, which is the first and only bNAb to be brought to large-scale efficacy testing. And I'm going to describe a little bit of its clinical journey. It was found, just like the Ebola monoclonal antibody was found, by looking at a patient. And this patient was a controller who was able to control HIV for about 15 years, not on antiretroviral therapy. And so the antibody gene was isolated, it was put into cells, it was produced in that process that I described earlier on that schematic. And then it went into further testing, and that happened in the group that I am with. And so when HIV VRC01 was put into people, it was found to be safe. There were no anti-drug antibodies. That is to say that the body did not neutralize those antibodies, and it was allowed to remain effective. It had a half-life of about 15 days, which is on par with most IgG antibodies. And when it went into patients that had HIV virus, it was biologically active and was able to reduce their viral load. And then it was advanced into efficacy testing in the antibody-mediated protection, or AMP trial. And I'll describe some of that. But I do want to look at these graphs here, because you're going to see a number of them and really just point out what you're seeing. So on the y-axis, you're looking at essentially the amount of antibody, and the x-axis is the time. And so when you infuse the antibody, it naturally is at its high point, and then it drops down. This right here was another infusion of the antibody. It went up again, and it went up very quickly, which is an indication that there is not anti-drug antibody that's binding it and preventing it from working. And then it has its slow trough period that it goes down. If instead you give the antibody subcutaneously, it's injected, there's a period of absorption, and then the same sort of pattern goes: a second dose, a period of absorption, and then a period that goes down. And this is the general pattern for how passive immunity works. You give a lot of it in the beginning, and it wanes away. And how it wanes away is very clinically relevant. Now, the AMP trial that I spoke to is admittedly a mixed result. VRC01 did not prevent HIV-1 according to the statistics that the trial was designed with when it was compared to a placebo. And what I will point out is there's no real way to claim that the trial was somehow not run effectively, and maybe VRC01 can do it, it's just the trial didn't show it. No, that's not the case. This was done in thousands of people in sites all across the world, and the retention of the subjects was incredible. 95+ retention in the trial, where these people were getting infusions of VRC01 every 8 weeks over the course of years. It really was a monumental effort. All of that to say is I think you can really trust the results very well. And when you look at the results, is that the cumulative incidence of infection in the placebo group is here on this line. And yes, the VRC01 groups were lower, so there was infection prevention, but not to the degree that would truly be necessary to claim success. And then when you look at it, you ask, well, how did they get infections? What type of virus were they getting infected with? Because HIV is not one thing. And what you find is that HIV-1 isolates, which were sensitive to the actual antibody, that means the antibody could do its thing, was actually very good and was able to maintain prevention efficacy in the 75% range. The problem is that many of the isolates, which are more difficult to neutralize, were a true challenge for the antibody. And so VRC01 was capable of handling some viral isolates, but not more. And if you're going to come up with a product that you intend to put into people to take care of a wide variety of the subspecies of HIV, you're going to need to do better. And this was, on the one hand, acknowledgement that we didn't have a product here, but we never really thought so. This was going to be proof of concept that an antibody can do anything meaningful at all with HIV. And this right here, this is the line that says, yes, you can, but you're going to have to do better overall. So how can you do better overall? Well, really, there are two broad ways you can do this. You can create a better antibody that simply does, by its very nature, the job better than others. And the second is you can make more of it, give more of it, and have it stay around for a longer period of time. And this is one tactic that you can do from the quantity perspective. So it's quality, quantity, and I'll talk about quantity first. And this is how all antibody recycling works. You have antibody in the blood, your IgG. It gets taken up into an endocytic vesicle, and this could be on endothelium, it could be on a number of areas. And then you have this purple here, which is the neonatal FC receptor, and it is just the same thing that is found in the placenta that will take antibody from mom, convert it over to baby. Well, the reason that happens is because the antibody binds here. And this is in an acidified environment which promotes the binding. And then you have separation, and any sort of proteins which are not bound will then go forward and be destroyed, whereas those bound ones will go off, and when they're in a non-acidic environment, will release from the neonatal Fc receptor and go about for recycling. So if you can increase the binding affinity for these receptors, then what you do is preferentially you take your monoclonal antibody and it will go through the recycling pathway instead of the degradation pathway, and that will therefore create a longer half-life. And the way that you can do that are specific mutations in the Fc region of the antibody. So that's the region that has effector functions, but not any binding. So you're not changing the binding, you're really just changing how this process over here works. And the way that you do that is you can take this amino acid, change it to a leucine, you can take this amino acid, change it to a serine, and then all of a sudden you have better binding and you have better half-life. And so we did that. And we never thought that VRC01 itself would be a product. So in a process happening in parallel rather than series, even before AMP started, we created VRC01LS. And we tested that in a phase 1 trial. And here are the results of that PK that I showed you before. And this black line is the exact same line that I showed you previously, where it gets infused, it goes down, you get another infusion, it goes back up, and then it wanes away unless you give another dose. The exact same dose of VRC01LS given once extends all the way out here. And before, whereas the x-axis was in days, it is now in weeks. And so you can see how much antibody you can actually give, where the half-life is extended from 15 days to 71. And a 71-day half-life is pretty incredible. So if you give a 40 mg per kg dose, you can stay very high for a very long period of time. So it's not inconceivable that you could have a twice-a-year injection of a monoclonal antibody that could protect you from HIV, which would be pretty incredible. So that's how you extend half-life. That's how you address the quantity problem. The way you address the quality problem is by going not to the Fc portion of the antibody, but to the Fab region of the antibody and actually doing some engineering or different antibody discovery, which can be more helpful to you. And so I'm going to talk about this part on the left here, which is VRC01. And what you're looking at is viral IC or inhibitory concentrations. And what you're looking at is how much antibody do you need in order to neutralize an antibody? And all of these, neutralize a virus. And all of these dots are different viral subspecies. And these viruses down here, you need very little of the antibody in order to create 80% inhibition of it. Now, if you go up here where you need very high concentrations, all of these dots are essentially viruses that don't get neutralized at all. And so what you can see with VRC01 is that there are a number of viruses that just plain don't get neutralized by it. And then you need a certain amount of it in order to neutralize the rest. You take a different antibody, VRC07523, which sounds more complex, and it is because it's been specifically engineered. So there are 523 iterations of VRC07, and this one was launched forward. It's capable, instead of neutralizing 87% of viruses, it can neutralize 96% of them. And when you look at the amount that you need in order to do it, it's much less. And so at the end of the day, you have, in theory, an antibody that can neutralize more virus, and you need less antibody to do it, which is pretty helpful when you're considering about creating a product if you don't need to give a lot of it in order for it to do its job. If you take these two antibodies and you compare VRC01LS, which is the same Fab region as VRC01, with VRC523 or VRC07523LS, you note that there's a shift of the probability of protecting non-human primates against a challenge model, because the VRC07523 needs this micrograms per ml, 0.47, to inhibit 50% of the infections, whereas you need 2.54 micrograms per ml of VRC01-LS. So they're both biologically active. It's just that the different potency of VRC07523-LS allows you to address more viruses with less antibody. And so you have to ask the question, well, what are you going to advance forward with another trial? If you're going to create another AMP trial, do you do something like VRC01-LS, which has actually a better half-life than VRC07523-LS? So if you look at 3 different antibodies, you have the VRC01-LS, which is superb with its 71-day half-life. You have VRC07523-LS, which has a half-life in about 35 days. And then you have the original parental VRC01 that has that 15-day half-life. So which is better? And at the end of the day, you want to neutralize virus. So do you take a less potent antibody, but more of it? Or do you take less of a more potent antibody? And when you come up and you test it against different viruses, what you find is that at least for some of them, even though you have about 4 times as much VRC01-LS as VRC07, the viral neutralization is greater for VRC07. And so if you do statistical modeling over time, the predicted neutralization is the solid bars of the different monoclonal antibodies, and their serum concentration are the dashed bars over here. You find that even though VRC01-LS lasts longer, the neutralization is much higher for VRC07. And that happens in a clade C viral panel, and it also happens in a multi-clade beyond just clade C. And so you can see over a period of time, VRC07 would probably be more advantageous. There does, however, come to be a cross point where you lose the effectiveness of 07 and you'd have to give a second dose in order to outpace VRC01. So these are how we kind of think about the balancing of quality and quantity. And so I'll just say, here we are. In principle, passive immunization against HIV may be possible. It will be very difficult, but even if you find that monoclonal antibody, or you find that combination perhaps of monoclonal antibodies, you'll still have difficulties with certain operationalizing of this, which will be, how many do you have to use? How frequently do you have to use them? What's the cost of goods to make this economically viable? Do you have to give this by IV, or can you use subcutaneous or intramuscular dosing? And then, as we've all seen with vaccines, do you have community acceptance of this? Because if you create the best vaccine in the world, if it stays in the vial, it's not going to be very helpful. And, you know, the true value in this, I think, is to use the monoclonal antibody experience, find out why they work so well, and then design an antigen that you can turn into a vaccine so that people can use this vaccine to create a similar type of antibody on their own, so it doesn't have to be passive. That's the general goal. That's the general outline of the project. And I look forward to the HIV world trying to push this forward. So at this point, I am going to pivot on to malaria and take these themes that I've talked about with Ebola and HIV forward. But I want to summarize some elements before I do. And the first is, it's not enough just to identify an unmet medical need and make any monoclonal. You really do have to decide whether the pathogen itself is amenable to antibody-mediated immunity, and if so, how. You also need to identify whether you're making a treatment, which is easier, as in the case of Ebola, or you're dealing with bona fide passive immunization, in which case it's much more complex and much more elaborate a task. And then finally, after designing the product, you're going to have to figure out, how are you going to test it? And I hope to show that all of these different steps are being taken and have been taken in order to address malaria as a pathogen. And the remainder of my talk will focus on malaria. And so rest assured, malaria is a true unmet medical need, especially in low and middle income countries across the world, specifically in sub-Saharan Africa, where there are millions upon millions of infections and about 400,000 deaths, mostly in children, and that's every year. And they're clearly at high risk, and there are clearly barriers to vaccination in just the pure science. There's pre-existing immunity. So, one of the reasons why people might not recognize why the MMR vaccine is given to children around 1 year of age is not because their immune systems can't handle it because they're immature. It's because the antibodies that mom gave to baby are so good that they will actually neutralize the vaccine and blunt its response. And so you need to wait for those maternal antibodies to go down before you can give the vaccine and expect it to have full immunity. And the same sort of thing can happen with malaria. And yes, there is bona fide immune immaturity with children, and that will be very important to determine their cellular response to the vaccine. And with malaria, it's thought that T cells are actually very important. Now, I'm going to show that I think antibodies play a role. Now, RTSS and AS01 as an adjuvant is a vaccine which is currently in use now. It does confer a 40% reduction in malaria, which is a good thing, but clearly there's mode for improvement. And it highlights that vaccines are not all equal. It's maybe an unjust comparison, but I think that RTSS is more of a flu shot than it is an MMR to deal with this sort of a pathogen. And so malaria as a parasite, I'm only going to be talking about Plasmodium falciparum as the target here. It does, of course, exist in multiple types of species that can infect humans, but we're going to be dealing with falcip because it is the pathogen that causes the grand majority of those infections and those deaths, especially in children. And so I'm going to ask you to think about this parasite, which is very different from a virus. And think about how Ebola was absolutely coated by that glycoprotein. And in any sort of effect of antibody, you're going to have a lot of antibody in order to coat the entire surface. And the target that we think is a good one is the circumsporozoite protein, or CSP. So the circumsporozoite protein of Plasmodium falciparum is going to be PFCSP, and it's the green that coats the entire outside of the parasite. It's very complex. It is over 2,000 transcriptionally active genes that go to make it in its native conformation. It's the most abundant surface protein, and it helps the development inside the mosquito. It helps motility inside human skin, and it helps with invasion of hepatocytes. And so it's a tremendous vaccine target. And here's the life cycle of how this actually works. Now, there is a separate life cycle all within the mosquito itself, which is left out of here. But you have sporozoites, which in about 1 to 3 minutes are in the skin. They very rapidly go into the liver, where they are replicating in their life cycle for about a week. And then they really burst into the circulation. They infect red blood cells. They have their own unique cycle. They create gametocytes, which then go back into the mosquito, which creates their own unique life cycle, and it goes on and forth. And so, are we making a vaccine for malaria, or are we making a treatment? And well, really, the target is a preventative vaccine with malaria, because once a person is clinically infected, the amount of myrizoites in the blood is tremendous, up on the order of millions. And really, I think it's just too strong a task to ask monoclonals to be able to handle that load, I think. I'd love to be proven wrong, but I think that the easier task here is to create vaccine like environments, which could even be used for elimination campaigns. And so again, we've targeted a preventative monoclonal. Well, what's the evidence that antibodies are actually going to be helpful when it comes to preventing malaria? And I'll show that anti-PFCSP antibodies do prevent sporozoites from infecting the liver at least 3 stages. And the first is in the skin. You get a bite from a mosquito, which typically can inject about 100 sporozoites into the dermis. About half of them will die. About 15 go to lymphatics, and they also die and do not create infections. But about 35% will go into the vasculature. And once they're there, they infect the liver in a matter of minutes. And once they're there, you can have sinusoidal egress, hepatocyte transversals, and then they establish infections and go out into the circulation again, and then infect the red blood cells. And so at all of these points, you have the opportunity for an antibody to intervene. So mAb114, Ebola, it came from an Ebola survivor. The HIV monoclonals, they come from infected patients. This monoclonal actually came from a phase 1 trial vaccine recipient of an investigational vaccine. And it's the next antibody that I'm going to talk about extensively. And so it's pretty remarkable to me that you can look at antibody levels for this phase 1 trial for the vaccine. You can look at the different dose groups. How much irradiated sporozoites did they get? Did they get 10 to the 3rd, 10 to the 4th, 10 to the 5th doses? Did they get 4 vaccinations or 6, 4 or 6, 4 or 5? You can plot their antibody levels, and I can tell you which subject we actually used to get the new antibody, which is titled CIS43LS. It's that one right there. I know that person. And you can then do functional assays and find out, well, which of these antibodies that they made actually can prevent sporozoite invasion in vitro models, and there's that person. And so this person, you can then take their B cells, find out which one of them is creating the antibody that's doing that, and create it in the lab. And so this is what we did. And it is the antibody named CIS43LS. And I'm going to talk about some of the features of it. And just like mAb114, it was extensively studied and evaluated in animals as well as in vitro prior to a go decision to turn it into a clinical product. And probably the feature that's most unique and exciting is that if you take the protein structure of CSP and you just plot it out in a linear model, there are regions of the CSP protein, and it binds right here. It's very unique. It's MPDP regions. And no antibodies had really been found that target this site before. They usually target all of these NANP repeats. That's both vaccine recipients as well as people who have natural infections. But this is where it is. And the exciting part is that this site is conserved across multiple PFAL-CIP field isolates, which means you're not going to get mosquitoes that are going to be harboring parasites, and those parasites are immune to your particular antibody, which happened with HIV, right? So you could neutralize some of the isolates, but not all of them. With this monoclonal antibody, you're essentially going to be able to hit them all. And when it was put into mouse models, it was able to really effectively prevent infection. And so, here's the overview of what we wanted to do. This is a schematic of a phase 1 trial that I was privileged enough to lead as the principal investigator. It was a pretty traditional model. We were going to start with a low dose, a 5 mg per kg group in both IV and subcutaneous routes. We were going to then dose escalate to 20 and 40 mg per kg. And then we were going to have negative controls who didn't receive any antibody. And then we were all going to challenge them with malaria. More on that later. And then we were going to take that data to do a part B to prospectively test a larger sample size with what we found in that different part. But unfortunately, that was what we wanted to do. Now I'm going to tell you what actually happened, because we had to pivot tremendously according to COVID-19. So, we started this trial. This right here is January of 2020. We dosed everybody, and we're going along, and right around this time period, we were going to challenge people with malaria, but we couldn't because of all of the restrictions that were put in place due to COVID. So it broke my heart, but we canceled that challenge. And you'll note that there's not a lot of sampling of different time points around here, because it took till about 20 weeks after dosing to really bring people back because of COVID. We got the samples. We were able to learn important things. We were able to learn that CIS-43 was safe and well tolerated in everybody. We knew that very well right away, but we were able to get the half-life, and it may not be 71, but a 56-day half-life is great, and it shows that the LS mutation, which is part of the antibody, is still active. And so, the journal really hated this consort diagram to show trial flow, but I still like it because I think it shows how elaborate everything was. These different boxes are the different groups of recipients, and 9 months later, we took them forward into an amended Part B. And, we had one person from the original group. We had one person from this original group, 2 people here, 2 people here. We had some others that we newly enrolled and brought on board. But this was a mammoth undertaking. And if there's any questions, I'm happy to talk about that. But we were able to go forward. And this is, a critical, crucial point. So again, you're looking at antibody levels over here. You're looking at time. You've already seen a lot of this graph, but here we took some of the original participants, we reinfused them. We had some of new participants, and these are their numbers. And I want you to focus on group 8, where there were 2 of 6 people from Part A who got the monoclonal back in February and did not get any new antibody. And then they were challenged on October 20th of 2020, because there's a serendipitous role here. And none of that would have happened without COVID intervening. And so this is kind of, if I have a joke, this is it. What's the difference between malaria, HIV, and Ebola? And the answer to that joke is the FDA will let me try and give you malaria. And so I'm going to talk about how you actually do that, because it's pretty incredible. Here you have mosquitoes, and these mosquitoes are at the Walter Reed Army Institute of Research in Silver Spring, our collaborators, and these are filled with FDA-approved parasites that we know are susceptible to our anti-malarial drugs, and they are waiting to bite people. And so you take people who have received your monoclonal and people who have not, and you place the cup over their skin right here, and you let the mosquitoes bite them. And the fun fact is, if you've showered or you've used soap or anything like that, they don't like to bite as much. So we ask people, not to shower or use deodorant, and they come in very early in the morning, so that's when the mosquitoes like to bite, and then they get bit. And then you take them to this room here, and this person will kill the mosquitoes, take them out, and dissect them under the microscope and see 2 important features. Now, what are they doing? They're finding out, one, did the mosquito feed? And so you have to see blood in their digestive tract. And then you have to find out how many sporozoites were actually there. And so you need to have 5 qualifying bites where you essentially have a lot of sporozoites involved. And so in this particular round, you had 5 mosquitoes in the cup. When dissected, these mosquitoes didn't have any sporozoites. This one didn't feed. And so you had 2 qualifying bites. So you go back, you make a new cup, you fill it with 3 mosquitoes, you let them feed, and then you go back and you find one of the 3 ate and had enough sporozoites. This one didn't have sporozoites, this one didn't eat. So you give 2 more. This sporozoite load was high, this was low. So you've had 4 of the bites. You go back, get another cup with one, and lo and behold, there's your fifth bite that has a good load. So that person is presumably infected with malaria. And because of the life cycle of malaria, you let them sit for about 6 days. And then you start doing daily PCRs for malaria, so you can detect malaria even before people are symptomatic. And you do that out to day 21. If they're infected at any point, then you treat them. If they're not infected, then you let them come back. And then on the final day, you give them Malarone in order to treat any infection which may be latent. And then you test for cure on day 49. And this is the tremendous punchline, is that CIS-43LS prevented malaria infection in everybody who got it. And so here's 100% of the CIS-43LS recipients in this experimental medicine model were protected. And the control subjects who did not receive CIS-43LS, well, 5 of the 6 were infected, which is what we wanted. Sorry, 5 of the 6 were infected, which is what we wanted. And again, back to that group 8, the people from January who were given 40 mg per kg IV, and then 9 months later were challenged, they too did not get infected. And so they had levels in their blood of around 50 micrograms per ml, and that protected them. And I never would have assumed that. If you looked at the mouse data, it would have said that you needed about 200 to 300 micrograms per ml to protect somebody, but it could do it with less. And so that was pretty astounding to me. It did, however, ruin my calculations, because what I wanted to do was to find out how much of the antibody was needed in order to prevent infections. And to do that analysis, you need to have exposed people to the antibody actually get infected. And because none of them were infected, we couldn't run that. So I still can't tell you statistically what number is needed in order to protect people. But it ultimately doesn't really matter, because I wanted to bold and underline that it was in an experimental model that we did this. But it really only matters whether it works in Africa or other places. And so NIAID is conducting a placebo-controlled trial in Mali, where it's given, CIS-43LS is given to Malian adults in the malaria season. And the data will be unblinded soon. And we will find out whether Kasum Kayantau's group in Mali and Peter Crompton at NIAID were able to really advance this, not in an experimental medicine model, but in a way that might actually help people in Africa. And so, lessons from this, serendipity produces success from failure. So, we produced this antibody out of a quote unquote failed vaccine. And COVID-19 could have derailed the entire trial. But because we had those people from January delayed until the fall, we were able to show how durable it was, which we never would have been able to do otherwise. We were also able to find a new site of vulnerability at that junctional epitope, and that could be used for vaccine designs. And then, just like VRC07 is a better example of VRC01, we have L9LS, which is a better example of than CIS-43, and that we recently completed a phase 1 trial on, and that manuscript is submitted for review. And so, what are my concluding remarks? Well, all vaccines mimic the native immune system, and so therefore understanding the native biology can lead to interventions for both vaccine and mAb designs. I am leaving the VRC in about a week to go on, and I want to say in my last public address here that I am beyond grateful for my mentorship training and early career here. The teamwork has allowed us to overcome incredible challenges, and truly cutting edge science can ultimately improve public health, especially in low and middle income countries. And these are the places that are disproportionately infected and affected by HIV, Ebola, and malaria. So I thank you for your attention, and the thanks are many to every one of these people here, especially the clinical trial volunteers, without whom we could do none of this work. and I'll answer any questions in what time allows. So thank you very much.